Hello, welcome to the lecture 6, so which is analysis of specialized metabolites, tools and techniques. So, what I have discussed in the previous two classes about the production of specialized metabolites in cell culture and in organ culture, root culture, and how to uplift the specialized metabolites in the cultures by treating with several chemical or biological molecules. So, and then also in the lectures I have shown the detection of metabolites. So, in this lecture basically I will talk about that what are the tools and techniques used for general detection. Let us go to the next slide. So, the things what I am going to cover here is basically routine phytochemical procedures which are used for extraction of natural products. Then we will talk about high performance liquid chromatography. We will talk about gas chromatography, mass spectrometry. We will talk about metabolite profiling, metabolomics and metabolite fingerprinting. We will talk about metabolomics platform and outline. So, once the plant material is collected, say it is whether it is from the field source or from the tissue culture medium whatever. So, that needs to be processed before metabolite can be extracted. So, it may be stored in alcohol or it may be dried and then it is extracted, it is macerated and then extracted with different organic solvents. And once the organic solvent, once it is treated with organic solvents, incubated and shaked for several hours, and then this will be further the solvent needs to be dried up, and this is an equipment called rotary vacuum evaporator, where the low temperature boiling will occur. So, the solvent will be separated from the natural product. So, solvent will be collected here and the natural product will be remained at the bottom of the flask, and this solvent can be redistilled and reused for another set of extraction. That is one way. The other way is that once you have the natural products, so you can do other thing also, like for example, this what we have shown that it is normal drying, but then the material can also be freeze dried, and freeze dried material can also be used for extract, or in the other way that once we have this materials here, then this can be, if it is more of polar nature, then it can be extracted with ethanol water, and then ethanol can be evaporated and the water phase can be made to ice, and then that can be kept in the lyophilizer, and eventually you will be able to recover the metabolite in the powder form. And one good example is the tea polyphenol. So, the tea polyphenol can be extracted with ethanol water, and then ethanol can be evaporated in the rotary evaporator. The water phase can be dried out, can be made to ice, and then that can be kept inside the lyophilizer, and then the material will be dried, the ice portion evaporated away without going to the liquid phase, and eventually we will see the powders of tea polyphenols accumulated in the vessel. There are two simplified glass equipments or glass apparatus which is used for extraction. One is called Soxhlet extraction. Suppose if we want to extract the carotene compounds, color compounds, say from flowers, say from fruits, say from dried fruits whatever. So, that can be extracted by putting the samples here, and we have to put the non-polar or even semi-polar solvent, and then like acetone, or we can use non-polar solvents like hexane, nitromethane or whatever, and then it will be refluxed here, and it will make repeated extraction through siphoning, and all the what the materials kept here, that the extracted material will come here, and later this material can be again rotary evaporated. So, the solvent will go away and your concentrated material will be at the bottom of the flask. Now, if you are planning for making essential oil extraction, from plant sources. So, either fresh or the dry plant. So, this is one equipment which is called Clevenger type apparatus, which is used for laboratory scale extraction. So, this is nothing but it is used for this hydro distillation. So, the samples are kept here along with the water, and then it is being boiled, and the steam generated. So, that along with the oils it will basically go up and then go up in this way, and then here there is a condenser, it is cooled down. So, the water and the oil both will come, and if the oil is lighter than water, so it will float above it like showing here, and then this can be collected later. So, this is the one example of one essential oil which is extracted in our laboratory. So, the hydro distillation has several advantages. So, like it is used for the extraction of the essential oils, and it is really very cheap technique, no solvent is required, and then you can go for preparative scale extractions as a very clean extract, high enrichment factor is there. The disadvantage is that quantitative distillation is time consuming and it is unsuitable for milligram scale operation, and also it is a possible of decomposition due to the presence of water and high temperature. So, that has to be taken into consideration. So, here there are some real examples. This is called allspice plant, which is dried leaves are available in the market. So, it gives is used as a flavoring compound for the food, and this allspice accumulate lot of essential oil, and this is basically the essential oil which is isolated through this Clevenger type of apparatus here, and this is ekangi. This is a type of ginger which produces this rhizome, and this rhizome produces the oil, essential oil, which is very fragrant, and that has also been isolated by this Clevenger apparatus. And this is the lavender plant. Lavender plant grows in the temperate region of the country, and there is a huge promise. So, CSIR, that is the Council of Scientific and Industrial Research, they actually floated a big, they are rather running a big program on aroma mission. Under aroma mission, they are making this essential oil extraction technology popularized, and this is one such plant which is installed in a village in Jammu and Kashmir, and this is the real extraction going on. You see the fire here, and this is a big vessel which contains the lavender material, and the other way the oil will be coming here and collected in the bottle. Now, that is a general way. Now, what about the specialized analysis? Like when we are talking about the drug plants, so and we produce that in the tissue culture, we elicited them, it is producing in higher amounts. So, it is important to see that whether the tissue culture materials are producing the metabolites, or whether the elicited materials are producing the metabolites, and can we really distinguish the difference in the content is elicited and unelicited. The best way to do that using the high performance liquid chromatograph. The equipment assembly is called high performance liquid chromatograph. So, where there is a pump, this could be a single pump or binary pump, okay, and this is HPLC solvent. So, solvent, it may be a mixture of 2 solvent, there may be 4 bottles, 4 solvents can be mixed it up and pumped here, and here is the injector where samples will be injected, and then eventually it moves into the column. So, where the separation occurred, and here is that the stationary phase, that is the column material is non-polar, and the mobile phase is polar, and the compounds are separated depends on its interaction with the non-polar phase. That is the C18, this could be C8 or it could be C35, depends on the type of analysis you do. For phenolic C18, for carotenes we go for C35 columns. C8 columns are also there for conjugated phenolic analysis, and then this will be separated depends on its interaction with the non-polar stationary phase, and then samples will be eluted out, and that will be detected in the detector, and the waste goes out, and this detector is connected with the computer system, and you will get the peak each peak represent one compound, and this is the real example of one such HPLC equipment available in our lab. So, here there is a pump component and there is a detector component, and these are the solvents, and everything is integrated here. It is next connect to the computer. So, this is the inside of the pump tubing, and this is basically the detector. It is called diode detector. Normally, there are several type of detector used. So, the HPLC in principle will work with the spectrophotometric principle. So, the most common detector is UV vis detector. So, diode detector is basically the advanced detector, where in normal UV vis detector you can detect maximum up to 5 different wavelengths at a time, depends on the capability of your detector and the software. But in diode detector you can detect the compounds say starting from 200 to 400 or 600. So, that means that every wavelength the compound takes the data and then it processed and finally, it forms a spectra. So, advantage is that in normal UV detector you cannot get the spectral information. So, you have to collect the elute and then you check it, run the spectroscopy in a spectrophotometer. But in diode detector it is basically online detection. So, once your samples are eluting out, you can always click it and you can get the spectral information. So, what is showing here the standard of chlorogenic acid and the sample. So, this is basically the peak overlay, from there we identify. Why the spectral information is important? Because particularly for the phenolic and carotenoid compounds, it is possible to identify the compounds based on the spectral information, and this will be compared with the standards, and once it matches we can identify, and certain deviation in the spectra can also be interpreted. It depends on its conjugation with another compound. So many things can be worked out with a diode detector. So, that is why earlier time it was called the poor man's LC MS. So, because MS detectors are expensive, that way diode is reasonably cheaper. Diode detector can also be used to analyze the spectra, and from there one can interpret the type of the metabolite accumulated. And what is showing here is a typical HPLC run. This is the baseline, this is called retention time, and these are the 3 other different compounds which are eluted after the column. Okay. And this is again another old picture. So, here is a old HPLC system and with a computer, and basically this is UV vis spectral analysis. So, this is like for hydroxycinnamic acid esters, how it looks. This is basically the flavonol glucosides, and these are the different HPLC peaks, and from these peaks the spectral information can be generated, and from there the compounds can be identified. Now, the similar tools was used in the laboratory for detection of metabolites. You are familiar with this because in the earlier class I have shown this. Now, it is clear to you that the red one is basically the HPLC profile of the control, okay, and where you see that the compounds are interested there, and the black one is the elicited, that means treated the culture with extract, and then after elicitation and control both the sets were extracted independently with solvents. Say it could be methanol, it could be ethanol, it could be other one. So, here it was methanol was used, and then it was centrifuged, and then it was concentrated, and then it was filtered, and then it was injected in the HPLC, and specific wavelengths were set for detection of the metabolites, because these are xanthone. So, it was kept somewhere in the UV region, also in the visible region also there is also the xanthones can also be detected, and so from this it is now clear that why HPLC analysis is important and what is its uses for this specialized metabolite analysis, and it is also possible to quantify this, and you can get the real estimate how much your product is formed. Not only the detection of metabolites, suppose what happens that these metabolites are formed as a result of enzymatic reaction. So, that means the substrate A is produced is converted into the product say B, okay, and there is maybe an enzyme X is there. So, what is important that once we get more product in the elicited culture, we need to see that whether the enzyme activity is enhanced or not. So, what you need to do, that enzymatic reaction means that in a test tube you add the cell free protein extract, which is the source of the enzyme, you add the substrate and carry out the reaction, and then you check for the product formation. So, the first experiment should be that you run only your product. If you have the standard, you run the product, you get the peak here. Okay. And the second one is this, that you add your product, you add your enzymatic reaction along with the product. So, this is only the product standard, this is the product with the enzymatic reaction. Now, if you see this peak height and this peak height, this is more. Why this is more? Because this include a part of the plant reaction product, and both actually arrived in the same place. That means, the exactly this product which is the standard is basically formed by the plant. If it is different, then otherwise you could have seen a small hump along with a big hump. The small hump indicates the plant reaction product and big hump is the product available. So, but it is the same, that means, it is the correct product which is forming in the plant, and this is basically the reaction, real reaction, no standard product was injected. So, this is the reaction of product formed after 20 minutes. So, first one is basically the black one is the unelicited one and the blue one is the elicited one. So, you see that there is a difference in the enzyme activities, and that as a result of that it produces the product. So, basically in this one the benzophenone synthase activity was checked by putting two substrate benzoyl CoA and malonyl CoA, and it produced the benzophenone. So, this is the benzophenone standard, and with this is basically the standard benzophenone UV spectra, and this is basically the enzymatic benzophenone formed. So, these two matches, from there one can confirm that indeed such reaction occurred, and through HPLC based method it is possible to detect the enzyme activity. Acquiring such skill is difficult, sometimes it takes 6 to 8 months to standardize such process. Now, apart from this UV detector, there is another sort of detector available. Unfortunately, I am not putting any figure here, but it is called refractive index detector, which is basically called an universal detector, because UV vis detector or diode detector only detect the metabolite which contain double bond and this aromatic ring, but other aliphatic compounds or other sugars, anything it cannot detect, but refractive index detector can detect anything. So, that means it detect all compounds, that is the advantage, but what is the disadvantage? It has poor sensitivity. So, sensitivity is less compared to UV, and another point is this, that in case of refractive index detector, one has to use, one cannot change the solvent composition, the solvent composition should remain fixed. Okay. So, cannot be combined with gradient elution. Although I have not spoken about gradient elution here in this class, because that will take more time, but so here that way it is limited, and moreover the detector is fixed. So, you cannot do much changes there, and only advantage is that this is relatively cheap method, and once you compromise the sensitivity, it is possible to detect any compound in the HPLC, provided you standardize it properly in the column. Now, we move into the gas chromatograph. So, what is the advantages and disadvantages of the gas chromatograph? So, the disadvantage is that for volatile compounds, gas chromatograph is very nice, you can do any analysis, but if you look for non-volatile compounds, then you cannot detect unless you derivatize. That means, the non-volatile compound has to be made volatile by derivatization, and Advantage is that no solvent consumption is there, only gas is there, and sensitive universal detectors are available and columns are durable, but GC is a destructive method, you cannot recover the samples. So, it cannot be used for the preparative work, but also disadvantage is that supply of high purity gas is needed. So, this is basically a schematic diagram of a gas chromatograph. So, where you see that there is this is the column and is which is inside the oven, and the sample is injected here, and the sample injection port is there. There are carrier gas which will basically bring the sample will be injected carrier port. So, it is heated. So, it will make a gaseous form, and then that will be carried out here, and this column will be separated. Is a very big column, it can be up to 30 meters, and then it will be detected in the what is called FID, flame ionization detector, that is a universal detector. So, that basically will detect. So, here is a schematic diagram of a flame ionization detector. There is a flame here, and the column, the eluates are coming, and then it is basically in the presence of hydrogen flame, it is getting ionized, and then that is creating a signal, and that signal converted into electrical signal, and then that is being detected in the detector, and we can see the each GC peak that basically indicates each compound. So, details of which I am not covering, because then it itself it takes long time. Just to give you an idea what are the things happening. Now, in case of what I said, in case of volatile compounds, it can be straight away injected into GC, but if it is a non-volatile compound, suppose if you are interested to analyze the sugar or amino acid, then it has to be derivatized by using several derivatizing agents which are very costly. So, like MSTFA. So, okay, so it will be silylated, there is one typical example, or it will form this methylation, and then this form is becoming the ester form, and then that is the volatile form, then that can be detected in the GC detector. So, FID detector versus mass detector. FID is universal detector for organic substances, and its principle is based on the combustion of the analytes coming from the GC column in a hot hydrogen air flame. What I have said, that is the why this is called flame, the flame ionized, and it generated the ions and which are basically sensitively measured in the detector. Whereas, mass actually the column straight away connected with the ion source. So, where what happens that once the eluates are coming out from the column, that will enter inside the mass spectrometer chamber, and then there will be high energy electron beams. So, that basically will interact with the eluent, and then it is basically fragmented into ions, and these ions fragmented ions can be measured by either magnetic or quadrupolar field, and that is a very sensitive method, and by that way each compound has different fragmentation pattern which are detected by the compound, and it gives each ion chromatogram. Each peak represent one potential compound, and that can be confirmed tentatively from the mass spectral library available. Usually it is supplied along with the GC MS software, and which can be used to tentatively identify the compound. For the final confirmation, it is important to run the standards. Okay. So, mass is basically again a truly universal detector, but is a highly complex apparatus, is very sensitive and also very expensive, and a large database with mass spectra exist. So, and structural data vary with the ionization techniques, that is important, because even your sample preparation derivatization can also affect the product detection as well. So, that is important, and Okay, and this is basically the system. I will come to that. So, this is basically the what I have mentioned, the GC column oven. This is the injector, and the samples are run here, detected and separated, and these are the carrier gas for the mass. So, this is the same GC, but instead of this FID here, this is the mass. So, the transfer of the elements inside the ion source, where basically it interacts with the high energy electron generated from the electron source, and then it is getting fragmented, and then it passes through the lens. Then is a mass analyzer which detect the fragmented ions, and then it generates the signal and it gives the ion chromatogram. So, here is the sample enters, it is under the vacuum. This is ionization chamber, there is the electron supply and this electron collected, then it moves and it is fragmented, this is detected and then it is generated, and finally, you get the peak. Now, let me talk about the application, what we can do, like volatile compounds, like flower emits scent. So, and with emit scent in the day and in the night, it is possible to collect the scent volatiles from the headspace and then analyze in GCMS and detect the composition. So, this is basically the headspace, that is the area where the flower emit the scent, and if you put a glass flask, then it will be filled it with up, then the emitted volatiles will be there, and then if you add a column adsorption matrix, and then if you add a vacuum pump, if you start the vacuum pump, all the volatiles will be trapped through this, and in between, as you have the column or the adsorption matrix where the volatile will be adsorbed, and these adsorbed volatiles can be later eluted through organic solvents, and then you collect that one the solvents with the your volatile compounds, then you can concentrate it, and then you inject it in the GCMS, and in the mass it getting separated, and eventually you will find different compounds. So, and you can elucidate the composition of the volatile compounds. So, this is the headspace assembly in the field, and this is the headspace assembly inside the plant growth chamber, where temperature and light can be controlled, and we wanted to see whether it affect the scent volatile synthesis and release. So, this is again the essential oil. One is the first one is the emitted volatiles from jasmine jui, no, from belly, Jasminium sambac is belly. So, in emitted volatiles we have seen these clean compounds, and this is basically from the extracted floral extracts, where you will see multiple compounds. That means, there are lot of compounds which are not emitted in the flower, but retained, but when you make the solvent extraction, all compounds are coming out. So, why it is emitting few, why it is retained, that again gives a scope for studying the fundamental science behind it and the physiology and the molecular regulation. So, and this is more of the practical application, and it is also possible to detect the enzyme activity. That means, the volatile compound is produced as a result of enzyme activity. So, this is the substrate, this is the volatile compound, and TPS is the enzyme. So, it is possible to set up the reaction in the test tube, where you add your substrate, say GPP, you add the floral tissue or leaf tissue, which is the source of the enzyme, and then you do the reaction. What will happen as a result of reaction? These products are volatile, it will try to escape, but as this is the seal tube, it cannot. Rather, here basically this is called SPME, solid phase micro extraction fiber holder is there. So, here is a needle, along with the inside the needle there is a fine cartridge is there. So, that cartridge is exposed, and the cartridge will get saturated with the reaction product emitted from the reaction, and once it is done, and this is basically you are setting up this reaction at a particular temperature. Once it is done, then this cartridge can straight away go into the GC port, and then you can detect it. So, here is the control, here is the product formed as a result of reaction in the crude petal, and this is basically the standards. So, basically through this it is also possible to detect the enzyme activity which is occurring in the either flower petal or in the aromatic plant leaf, and as a result volatiles are produced. So, such sensitive and fine analysis can be done with the use of SPME linked with GCMS. And I have also mentioned in one of the previous classes that this is called biological detector, where that how much sensitive volatile compounds to the insect can be detected by connecting with an electroantenographic detector, where basically an insect's antenna is mounted here in a part of this detection system, which is called electroantenographic detector. So, here there are two electrodes, and here is the real antenna cut and mounted. So, this is basically the bumblebee. So, here is the antenna, and then it is basically put it here. So, once the metabolites, the floral metabolites or leaf metabolites, it is separated. So, there is a splitter here, one goes to the FID, which gives signal and get the GC trace. So, the other part of it goes here out, and here it is basically it goes towards the electroantenography detector, where the filtered humidified air is to be supplied, and this will be basically responded by the antenna. If it is biologically active, then it gives a peak, and you have to basically compare the peak here with here. So, out of so many compounds here, only 1 and 2 compounds, only 2 compounds for the biological activity. That means, out of this 6 or 7 volatiles detected in the flower, only 2 are biologically active, and this is basically the GCEID facility which is used for detection of this. So, so this is all about it, but then another 5 minutes I will talk about the metabolome analysis. So, what I have discussed so far is basically targeted analysis. That means, we are doing cell culture or elicitation or multiple shoot culture or even the floral volatile analysis. We are looking for targeted analysis. Okay. Now, it is also possible to see what happens as a result of say certain environmental stress to the plant, which may be untargeted, which we do not know. There are certain changes. If we make our mind set for target, we may not be able to find some of the metabolites which plays important role, but we simply ignore because of our, we simply avoid because of our ignorance. So, it is important to go for sometime the untargeted analysis. The untargeted analysis is rather more complex, but I will just give you. So, that world metabolomics, plant metabolomics world is coming to that. So, untargeted analysis, the first part is same, but it is important that you find a biological question, then only you go for untargeted analysis. Proper design is required. Then this part is same, tissue harvest, extraction, analysis. After analysis, there are lot of, after the GCMS run, there are lot of post run analysis, which includes this computational statistical tools, and eventually that gives the real life picture. Now, it is important to understand the concept like metabolite target analysis, where specialized protocols for analysis of difficult metabolites can be generated. So, in metabolite profiling means that we already have the defined targets. Whatever I have discussed earlier slides, those are basically metabolite profiling, but metabolomics means it is unbiased identification of all the metabolites present in a particular condition, and that condition perhaps arises because of certain changes in the environment or so. Okay. Or also you can do elicitation. We are looking for only upliftment of indole alkaloids, but we never know what happened in the status of the primary metabolites. So, that will be untargeted or semi targeted analysis. So, nowadays such study is also important to provide the holistic view of the metabolism occurring in the plant cell. And metabolite fingerprinting means instead of separation of individual metabolites, so it basically collect these metabolites together and class them in terms of crude mixture metabolites. So, that is presented in terms of fingerprints. So, their individual metabolites are not important, but the fingerprint pattern is important for identifying the particular sort of reactions or the group of metabolites present there. So, here is the single compound assay, here is the targeted analysis. here is the metabolomic analysis and here is the metabolite fingerprinting. You see the difference, single compound assay, targeted analysis, that is multiple things what I have discussed today. Metabolomic analysis is more complex, you will find lot of untargeted compounds and metabolite fingerprinting. So, this is basically an approach of metabolomics is followed. So, it involves both GCMS or HPLC or that is called HPLC-MS or we call LC-MS, MS. The important point is that once you generated the data, once you acquired the data in GCMS or HPLC, then there needs to be data deconvolution. That means, you have to clean up the data, you have to avoid the unwanted peaks, so that what you are looking for it makes sense, that is called data deconvolution, and then there will be a network computation and then you have the statistical analysis. So, here what is showing the targeted analysis, where you are looking for a particular compound in a wild type and say you are transgenic or knockout, whether that compound is enhanced or reduced, and untargeted, this is what is in the wild type. Okay. And when untargeted analysis means when you are transgenic or knockout, when you look for untargeted analysis, you found that peak number 3 has enhanced. In the targeted analysis, you never plan to look for that one, you plan to look for only one. So, but if you compare here and here, the 3 is more prominent, because you did not set your mind for untargeted analysis, you avoided, you basically missed that. That is why it is important to go for untargeted analysis to get the holistic picture of the science behind the metabolism. So, this is the more or less we are coming towards the end. So, this is basically the flow diagram of the metabolomics. So, like tissue harvest, storage, metabolite extraction, concentration, derivatization, resuspension, then targeted metabolite, untargeted metabolite analysis, use a range of equipment GCMS, LCMS, capillary electrophoresis, MS and MALDI, then data processing, okay, then data preparation, data normalization, then statistical analysis, multi and univariate analysis, then peak annotation, then functional interpretation with the network analysis, then metabolite mapping, and then finally, you integrate this data with other databases available for the plants like proteomics and transcriptomics, and eventually you will get the whole information. So, this is in brief the metabolic platform. So, analytical tools can be used GCMS, NMR or LCMS, raw data generated, then pre-processing, okay, which includes noise reduction, baseline correction, alignment, normalization, scaling, then chemometric analysis, which is peak, deconvolution, classification, regression, and ultimately you get the score plots and which help to interpret the biology behind some changes, behind that changes. So, integrated functional genomics, where basically you have the genome information, transcriptome information, proteome information, metabolic information, all things to be clustered together, and that will give you the wealth of knowledge for the biological changes occurring, and based on that the strategies can be designed for metabolic engineering or genetic engineering. So, that is all about it. So, it is important that choose the rationale behind specific methods, and the choice of particular methods it depends on the type of metabolites you analyze. Sometimes that method is good or bad, that is not important. It is important that what different methods basically provide you, and it is also important to optimize the system. So, that it is important that you should know the specialist skills, equipments and the resources whatever available and especially the time whatever available for the analysis of the metabolites in question. And with this I end this lecture. Thank you.